Hi there, everybody. I hope you're having a great day. But even if you're not, it's going to get better. You know why? Because it's time for Asynchronous Recorded Lecture 33 for Bio 145 Microbiology. This lecture is scheduled for Wednesday, April 15th of the year 2026, and the topic is viral viscerotropic diseases. Now, since this is a new topic, we're not going to have a recap. So let's just go straight to the new topic. So that does it for our skin ones. Now we're going to go to viscerotropic viral diseases. And viscera is like the insides, the innards. All right, that's the insides. And this is just a grouping name for viruses that don't fit into the respiratory tract or the skin, things like that. We're going to talk about two of them for this semester. I change from semester to semester. But this time we're going to do rotavirus and HIV. Rotavirus and HIV, most of you know that's AIDS. All right. So first, let's talk about rotaviral diseases. We're going to talk about gastroenteritis. Sometimes this is called the stomach flu. We're talking about an inflammation in the digestive tract, and we're going to focus on that as gastroenteritis, like nausea, vomiting, diarrhea, those things caused by a virus. We're going to focus on that which is caused by rotaviruses. Now, gastroenteritis can be caused by noroviruses and other kinds of viruses. They're often called the stomach flu. All right, but that's a bad name because number one, you're going to see these viruses infect not stomach cells but intestinal cells. And number two, these are not flu viruses. These are not influenza. These are rotaviruses. Okay, and rotaviruses, these look like wheels. All right, so here's some electron micrograph of rotaviruses. So we're just going to focus on GI tract, viral diseases, nausea, vomiting, diarrhea caused by rotaviruses. Those are the agents. But just be aware other things can cause GI viral diseases. So the agent is rotaviruses. They look like wheels. What's important? Well, they've got a double-stranded RNA genome. We're not going to talk about that because I didn't go over how those reproduce. What I want you to remember is they're naked. All right? They have a very strange life cycle, but they're naked viruses. So knowing they're naked, you got to know, they're going to cause lysis of the cells, right? The transmission is fecal-oral, okay? Meaning from the digestive tract of one person, the viruses are in there. They go out in the poop of that person, and somehow they end up in the mouth of the new host. The incubation period is about 2 to 3 days. So there's your 2 to 3 day rule again. The symptoms, nausea, vomiting, diarrhea. These can lead to just annoying diarrhea in some people, to really diarrhea that leads to dehydration, a thickening of the blood, and death, especially in the very young children, babies. It's also the immunocompromised. So it can be deadly for people. It can lead to death. It is extremely easily spread. So I'll always tell this story. Many years ago, there was a cruise ship, and there was an outbreak of gastroenteritis caused by rotavirus. All right, it was there, and so a lot of people got sick. People went off the boat. The cruise ship company scrubbed down the ship, took it out of action for a few weeks, really cleaned it well, and then put the next group of people on it. Unfortunately, they can't, again, another outbreak. So they're like, oh gosh. And so they decide, heck, we're going to scrub it down, keep it out of action for a whole year, and then we'll use the ship again. And interestingly enough, by the way, what they do, they rename the ship because there was such bad press. They decided, well, we can't send that out again. So they renamed the ship. After that, it was fine, though. There was no more rotaviral outbreaks. Well, why did that happen? Why did that happen? Well, it's a really hardy virus. The virus, it can last for like weeks to months on a surface. It won't, quote unquote, die out. So then maybe at months it's been on some surface, person picks it up, gets on their food, contaminates the food, boom, person eats it. Even though it's been outside the human body for months, that virus can still infect human cells. This is, a lot of people get this as very young children in daycare centers. Because daycare centers, people have to clean diapers, change diapers. Little kids aren't that sanitary, go out washing hands and stuff like that. So the virus can get out there on surfaces, and then food is prepared on those surfaces. Little kids lick toys that they've touched with their fingers after going number two. You get the idea. Viruses last a long time. Spread often daycare centers. So one thing, they last a long time. Secondly, it's very concentrated in poop. Okay, a person who has a really bad rotaviral infection, all right, they actually measured it, studied the number of viruses. And this was in an article I read, and I thought it was a typo, and I double checked, and this is a true number. And I usually, when we have the class, I play the auction game. And I have it guess how much they found. How many viruses were in a gram of this person's poop. All right? And well, we can't do it now, but here's an incredible number. This is worth memorizing. It's not just cocktail. 100 trillion viruses in a gram of poop of a badly infected child. So you're like, wow, 100 trillion. But then you're like, well, maybe if the dose is a million or a billion, okay, that's not too bad. No, it's a hardy virus, and it's also a very successful virus. The virus is only about 10 to 100 viruses make up a dose. So you can see all these things. It would explain why it can lead to outbreaks and be so hard to get rid of on a thing like a cruise ship or a daycare center or something like that. All those factors make this virus very successful in what it wants to do. So let's talk about the pathogenic mechanism. First of all, viruses, they infect the epithelial cells of the small intestine. They take over. So they come in on food. They have some level of they can withstand the acid in the stomach. They come in, they bind, and they infect those cells. And they're naked viruses, so you can imagine direct penetration. They go in, they take over. This is rare that you see in a virus, but they actually make a toxin. It's not just reproducing in your cells that hurts you. They actually make a toxin. It's called NSP4, and it was called a little cocktail. They weren't sure what it did. They called it the non-specific protein number 4. All right. And it ended up being a toxin that causes the loss of waters and salts from the epithelial cells. And it can be very, very severe. Great loss of water and salts. But so the poop becomes very watery, liquidy, diarrhea, bad liquidy diarrhea. But it's not bloody. Not bloody. So what does that remind you of, folks? Out there that are listening. What does that remind you of? What disease did we see that before? Was it typhoid? No. Cholera, yeah, remember. So, NSP4 has a similar outcome or effect as the cholera toxin. Massive loss of water and salts from small intestine cells. So think like cholera toxin. A little cocktail in third world countries. Look at the number of babies that die per year from rotaviral gastroenteritis, just because the diarrhea is so bad. Okay? So what happens? The cell lysis occurs. There's inflammation. Remember, it's a naked virus. And luckily, it doesn't lead to a carrier state. Usually the person gets over it. They don't become carriers. So they will eliminate it. They will be cured of it, which is a good thing. All right. The treatment, unfortunately, is just you wait it out 3 to 7 days. Usually it ends by then. You make sure the person drinks a lot. It replaces the water, the salts they've lost. That's pretty much all that can be done. There's no drug per se that's given to that person. Prevention used to be a real problem in the U.S. Okay, I've got this stat here that in the U.S. before there was a vaccine, there were about 70,000 hospitalizations a year due to rotavirus, mainly because of the bad diarrhea, primarily in children, in babies to young children. All right, there has been a drop in about 75 to 90% drop in the incidence because of the vaccine. There's 2 vaccines, but the major one is called the Rotatec vaccine. It's used in the U.S. Rotatec, and it's a live attenuated virus. So think it's live attenuated, means it's weakened. Remember, it'll reproduce in you. All right, won't hurt you. And what are you going to get? You're going to get a B cell response with antibodies, and it's living, so MHC1. So you're also going to get a cytotoxic response. and it's been a very successful, dramatically effective vaccine. There was this one article, one article led to another, and it talked about the economic implications, a little cocktail. And this article is basically saying that this vaccine, because it stopped all these mainly kids from going to the hospital, saved over $1 billion to the country in productivity. You're like $1 billion, and it's basically saying that when all these kids went to the hospital, their parents would take off work. All right, and they would, because they would lose work time. They lost the country lost productivity. So about $1 billion, over $1 billion. So this virus has saved the United States in terms of work productivity over $1 billion. That's what it claimed in this article. The main part of the article is also to basically say, companies should make sure their health insurance provides vaccines, because in the long run, the kids get sick less, the parents work more. Our next thing we're going to go to then is, now that we're done with our rotavirus, is we're going to talk to HIV/AIDS. And this is brief because, at like Davis, there are courses whole quarter long that talk about HIV/AIDS. We're just going to talk about part of a lecture, but that's what we have time for, and I'll do my best. A little cocktail about it. 35 million people have HIV, about 5 million die every year. Okay, very serious. The agent is human immunodeficiency virus. HIV, that's what it stands for. It is a retrovirus, so think it's an RNA genome, and that's going to be used to make DNA. It's transmission by human reservoirs only. Can't get it from chimps and monkeys or something else you might read on the internet. And the viruses are particularly highly concentrated in the blood. So here's just an example. In the blood of someone who's infected, you'll be at 1,000 per mil. While if it's a man, in the semen, it'll be 50 viruses per mil. In vaginal fluid, woman, 50 viruses per mil. So it's most concentrated in the blood. So that's why, you see transmission where there's blood involved. Okay, when there's blood involved, that greatly enhances the chance of transmission. So kind of keep that in mind. It's very important because that's where it's most concentrated. So let's look at the transmission of HIV. So following the idea, when there's tears in the epithelium, in the skin, tears, so the blood can get out, that's when you're going to see transmission is most highly successful. So if a person practices anal sex, where there's damage to the skin, usually there's bleeding. A person has vaginal sex, but there's STDs there. Well, sexually transmitted diseases. Think tissue damage, blood. All right? That's where you see the highest, most efficient transmission. So, homosexual men, anal sex, prostitutes having a lot of sex, high chance that they'll have some type of STD. There's lower transmission if someone's having vaginal sex and there's no STDs. Right? Are there other transmission means other than sex? Well, yeah, think blood. Okay, blood transfusions. Now they always screen blood supplies in our country for HIV. But early on, when HIV first kind of became apparent, came into the afflicted humans back in like the 80s, they weren't screening blood, and people would get it through blood transfusions. Mother's milk. Yeah, there can be quite a bit of HIV in the mother's milk. The baby, again, fairly immunocompromised. Needle share. You see this a lot. People that abuse IV drugs. Some cities will say, hey, yeah, you're breaking the law, you're abusing drugs, but here's clean needles because we don't want you to spread HIV from one to the other. Because sometimes people that are IV drug abusers, to save money, they'll reuse things, they'll share needles. So that's why you'll see these free needle giveaways. Can you get it from kissing? No. The concentration is very, 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 very low, so you don't have to worry about it in the saliva. All right, kissing. Okay? Let's look at this picture of our HIV virus, a little cartoon. What do we got here? Because we're going to be talking about all these things. Okay, again, it's an enveloped virus. I'm going to tell you right now, it's an enveloped virus. So what's it got in its envelope? We're going to talk about GP120 and GP41. They're kind of together, okay? Their proteins are kind of work together in the same spike. So that's going to be in the envelope. We have a capsid here. Okay, so here's the capsid. Looks like a tie, doesn't it? Got that outline of a tie. Okay, and what's inside the capsid? So what are the soluble proteins? We're going to have reverse transcriptase, soluble protein. Again, your human body doesn't make it, so the virus has to bring it in. And there's going to be integrase is another enzyme we're going to talk about. And there's protease. Okay, there's proteases. So let's kind of talk about that. And again, it's a single-stranded RNA genome. Sorry, I forgot to talk about that. Remember, but it's going to be used to make double-stranded DNA by a reverse transcriptase. So hopefully these kind of make sense as kind of a review. Okay, there you go. Keep this in mind. Well, so let's talk about the HIV life cycle here. It's an enveloped retrovirus. I'll go into the details after this slide, but it's an enveloped retrovirus. It's going to attach. It's going to enter by fusion. Okay, it enters by fusion. And the reverse transcription happens, meaning RNA is released from the virus. That's the RNA genome. And the reverse transcriptase, reverse transcription, uses the RNA single strand to make double-stranded DNA. The double-stranded DNA is then integrated. Okay, it's going to be integrated. It's going to go into the human chromosomes. So copies of the double-stranded DNA that were made from the RNA genome of HIV are going to be inserted into the human chromosome. So now we're talking about integration. Integration of double-stranded DNA, viral double-strand DNA into the human genome, human chromosomes. At this point, it's double-stranded DNA. They're viral genes. They're DNA. They look like normal genes. And your body's RNA polymerase is going to transcribe it. And your body's ribosomes are going to translate those proteins. And we're going to talk about the HIV protease. That's one of the proteins that's going to be made. The HIV protease is going to be made. And so all these things assemble, the viral proteins, the viral RNA, okay, when it's transcribed, not only is it going to be that RNA is going to be translated into viral proteins, but hey, that RNA, not only is it transcribed and translated, when it sometimes isn't translated, it is actually genome, and it'll go part of assembly, and it'll kind of come together, assemble with the viral proteins. And like I said, real important, HIV protease is in there, reverse transcriptase is in there, all right, integrase is in there, all those that were in, they're in there. Okay, they're going to be put in there, and then it's going to bud off. After assembly, bud off, and now it can go and infect other things, other cells. Just remember, does it bud off anywhere? No, it only buds off in areas where the parking spots are, right? Where the viral proteins, these newly made viral proteins, okay, some of them are going to go and insert into the membrane, the plasma membrane. So a lot of this stuff, it should make sense now. We've talked about a lot of examples of how viruses come together, made, they assemble, they bud off. Okay, it's pretty much this slightly different proteins involved, but same general pattern. It should come to you by now. Okay, go over it, get it. So let's kind of break this down into steps here. Okay, I went the overview. Let's go into a little more detail. So HIV, the disease caused by HIV, can be divided into 3 phases. Okay, so we're going to go through phase 1 first, and that's phase 1 is like when the person just gets infected. They have sex, or they use a dirty needle. Any way that the HIV virus comes into them, all right, that's initial thing is called an acute infection phase. Phase 1. HIV somehow enters the blood. All right, again, blood, from blood into blood. And now you say, well, what's involved? What's the ligand? The ligand on the HIV virus is called GP120. Go back to the cartoon a couple slides ago. GP120. And it binds what? The CD4 and CCR5 receptor combo. So you go CD4. I've heard that before. T helpers. Are we saying that HIV likes to infect T helper cells? Yes. That is the main type of cell. This is a T helper. This is the main kind of cell that is going to be infected by HIV. Now, there are some other cells, other leukocytes, but we're not going to get into those. Okay, let's just focus on what it does to T helpers. So it binds a CD4 and CCR5 on a T helper. Okay, and it's going to do fusion. Like I said before, it's going to fuse. All right, and it's going to uncoat. So once it uncoats, after it breaks open, again, it's brought in. It's reverse transcriptase and integrase. Those are soluble proteins. All right, it's not going to wait to make those. It needs those right away. Your cell doesn't. Human cells don't have those, so it needs them right away. It needs to make use of them. So they're soluble proteins. The reverse transcriptase takes the RNA, all right, and uses it to make DNA. So it goes from RNA. This is the RNA in yellow to double-stranded DNA. Now, I'm going to tell you that reverse transcriptase is sloppy. It often makes mistakes. Think missense mutations as it makes the double-stranded DNA. Kind of hold that. We'll get back to that later. Then the integrase, another soluble protein in there, takes that DNA, all right, shown in yellow here, that double-stranded DNA, and inserts it into the chromosome. So we went from RNA, genome, reverse transcriptase, a soluble protein in blue, makes a double-stranded DNA, a viral HIV DNA, integrase, then integrates it into the genome, the human genome. Okay. Now from there, okay, now it's DNA. Your RNA polymerase, guess what? It's going to transcribe it, and your proteins are going to translate that HIV mRNA. Everything's all set up to go, right? So there's a lot of trickery here going on. Let's try that again. So as I just indicated, the host RNA polymerase and transcribes that mRNA with that makes viral mRNA, shown right here. And it is then translated by the host ribosomes. And here we go. You're going to get your proteins. Okay? Because again, the cell's been tricked. The transcribed mRNA, what's it good for? Okay? Well, number one, that mRNA is just, again, translated into things the cells needs. All right? Now, some of the proteins are going to go into the babies. All right, so think, oh yeah, the integrase, the reverse transcriptase, we kind of talked about those. But also you got proteins that are needed right away, including the viral proteases. Because remember, when we go back, we talked about antiviral drugs. Often what happens is when the proteins are made here, okay, when they're translated by your ribosomes, they're made as fusion proteins, remember? So the proteins are all linked together in one long protein chain. And there's a viral HIV protease, which is really important to kind of break it up into individual subunits, into individual active proteins. All right? So the viral protease, it basically is made, all right? It's one of the proteins that's made, and its job is to basically chop up the big long proteins that were made, fusions that were made into individual active proteins. So viral protease, HA, is very important because without it, the proteins wouldn't be active. They'd all be stuck together in a six-pack. They need to be cut apart. So it's very, very important. It helps the protein mature, all the proteins mature. So you can imagine in the cell, you've the genome, RNA that was made acts as genome. RNA that made is translated into proteins. The proteins do their job, like viral RNA polymerase, and then, viral protease, and then these proteins come together with the RNA, and they assemble, and they can butt off, right? And like I said, the viruses butt off here. Again, they go to areas where they see, our parking spots. All right, there's some viral proteins. Think, well, what needs to be in the envelope? Because these are going to be in the GP 120, GP 41. Remember that? So that's going to be the part they're going to bud off, and now they can go and attach on some other cell. The cell, in this case, because it's being worked to death, it's going to die. Now, at this point, we're going to talk a little bit again about cells dying from syncytium formation. So as I explained earlier, the GP41 and GP120 proteins, they should be present in the infected cell's plasma membrane. So they're here. You can kind of, I'm just going to mark them off here. So this is an infected cell, and it's expressing its proteins, and there's proteins on the surface. These surface proteins are going to end up being in the envelope of the baby viruses, because the baby viruses are going to be pinching off, budding off here, correct? But those proteins are there to begin with, okay? They're going to be there. But what's interesting is GP41 has a function here in the plasma membrane before it becomes part of the envelope. Basically, GP41 is going to be one of those proteins that's key for fusion between an infected cell, the cell here, and count them, 4 uninfected cells in this example. Because GP41 is on the surface of this infected cell in the plasma membrane, it will trigger this one cell to fuse with these other 4 uninfected to make up a syncytium. Right? Where have we seen this before? HSV, yeah, herpes simplex viruses, and tuberculosis. So again, we see that theme of if it works in one system, it can work in the other. So you might ask, why does it do this? They're really not sure why. Scientists think that, well, this is a way for baby viruses, instead of having to bud off and be exposed to being attacked by antibodies, this is a way of baby viruses to go directly into new cells. All right, so the viruses that were in here, instead of pumping off, are going to be in here now. So it's kind of saved them taking a trip through the body fluids where they might be attacked by antibodies. But still, humans know. Nobody knows. But in this case, you can see that the human cells are going to die either from being worked to death individually or by forming these syncytia, these giant cells. So at this time, the person has flu-like symptoms. They don't realize they have an HIV infection usually. And these symptoms pass. They think they're perfectly normal because, like I said before, your antibodies and cytotoxic Ts are going to work to get rid of the HIV, whether it be in the body fluids with antibodies or those in the infected T helper cells as the cyto-Ts. However, the body never is successful in ridding itself of all the HIV. It never gets cured. So why aren't the antibodies and cyto-Ts completely able to wipe out during this acute phase? All right, why are they can't get rid of them? And the answer goes back to what I told you before. Reverse transcriptase makes errors when it first used that RNA genome and converted it to double-stranded DNA. Think of these mutations, single substitution mutations. If they're in coding sequences of viral genes, now you have missense mutations. All right, missense mutations. So think now the viral proteins, they slightly change in your body, which means, and if those are being used as antigens are changing. So it's not hard now, I think, to understand the idea that if you have a mutagenic virus, and HIV is extremely mutagenic, much more than colds, much more than flu. Basically, if you go from what a viral protein looks like in one of your cells to, so okay, let's say the virus, then new baby viruses leave, they attack other cells. Basically, if you look at the viral proteins in one cell of your body versus other, you'll see differences. So as it goes from generation to cells in your body of infection, the proteins are changing very rapidly. The antigens are changing very rapidly. All right? And your immune system just can't catch up to it. Now, in a flu, yeah, that happens. But you only see those changes if you compare the flu in one person to maybe a flu in another person who got that flu maybe weeks later. It takes a long time to see those changes. But in your body, HIV is so mutagenic. You see those changes in proteins. You change the changes in antigens within the same body. It's so fast. And that's why your immune system is always one step behind. Those antibodies and those cyto Ts just can't seem to get rid of all of them because it keeps changing its antigens. That is like why it's so successful. Now, like I said, things will get better and the person will recover and think they'll get over a flu. They have not entered the AIDS stage. That's the third stage. So they seem fine at this point because they've recovered from what they think. So remind me what happened during that acute phase. All right, during that acute phase, HIV came into the body. It reproduced greatly, pretty rapidly, and then your immune system started catching up and destroying it, the virus, your B cells, your cyto Ts, destroying it, and it almost went down to nothing, but it doesn't go to nothing. It seems to stabilize. That's at the end of the acute phase. There's still some there. If you're looking at the T helpers here, they got infected. The virus killed them, worked them to death. Syncytium formation. Your own cyto Ts killed some of those cells. But then pretty much the virus is almost wiped out, and now the T helpers start reproducing again, and they start accumulating, and they get to a fairly decent number. So now we're at the end of the acute, and we're going to begin chronic latency. So what happens in that? So welcome back to part 2. So we're discussing the pathogenicity of HIV. We've gone through the acute phase. Someone feels like they just gone through the flu-like symptoms. They've gotten over it. They seem perfectly fine. Now they enter the second phase, which is the chronic latency phase. Starts about, depends on the person, the individual, but about 2 months after the initial infection. All right. So think that at this point, again, someone's feeling perfectly fine. This name is really bad because, yes, the virus are latent. You're going to find out, but most of the time they're latent, but there's going to be these pockets of sporadic activity. And the word chronic just means constant, long-term. It doesn't mean like chronic, like you're feeling, like we said, oh, you have a chronic disease, you're feeling it. No, during this phase, the individual infected person is not feeling any symptoms usually. So it's kind of a misleading name. So as you'd suspect from the name, HIV is now going to go latent in the T helper cells, and it's known as a provirus in this time. However, there will be a sporadic activation of the provirus, so it becomes an active virus that starts replicating and killing the T helper cells. Now it's sporadic, so let's just say out of 1,000 T helpers in the body that have a latent infection, maybe 5 of them at one given time will suddenly wake up, become active, and trigger viral reproduction again, meaning there'll be transcription of the HIV genes by RNA polymerase. The RNA will be translated by the ribosomes, proteins will assemble, and new viruses will leave the cell just like before, just like we talked about before, just as in the acute infection, and the cells die. All right, but this happens 5 out of every 1,000. And then you go later, and then maybe 5 of another set of 1,000, this happens. But this just keeps happening. A few get turned on, a few get active, they cause death. All right. Now, during this time, the person doesn't realize it, but over this 5 to 10 year period where the person's basically asymptomatic, they don't have any symptoms, more and more T helpers are slowly being killed off. And the concentration is slowly, surely going down because of the sporadic activation and the result of it. Because again, the virus is going to reproduce, work the cells to death. The virus is going to trigger syncytia formation. The cells die. And also, this becomes a much more important factor now. The cytotoxic Ts that are killing virally infected cells, they're destroying what cells? T helper cells. And if you think about it, that's really bad. Why? because the trainer, the T helper cell, is being killed by its student, the cytotoxic Ts. It's like, here is the Karate Kid. Okay, everyone loves the Karate Kid, right? But the Karate Kid is being trained by Mr. Miyagi and turning around and killing Mr. Miyagi. So long term, imagine there's lots of Mr. Miyagis and there's lots of Karate Kids, and you need the Mr. Miyagis to train each generation of Karate Kids. If the Karate Kids start killing the Miyagis, pretty soon what? There's not Mr. Miyagi around. Pretty soon there's not enough T helpers to train the new sets of cytotoxic Ts. And you know, this movie ain't gonna end well, folks. It's gonna be sad. Very, very sad. So, you know the movie isn't gonna end well at this point, right? You figured that out. Because during this chronic latency period, the virus is still highly mutagenic, continues to avoid complete destruction by the cell-mediated immune system and the antibody-mediated immune system. The person still feels fine even though what's going on in the body, there's no noticeable signs and symptoms yet. However, this still is not at AIDS stage yet, not at the third stage. So thinking about it then, let's think about it. Let's recap what's happening in chronic latency. Well, slowly the T helper concentration, what's happening to it? It's going down, right? Going down. And it's about to reach this threshold, which we have an asterisk. When that concentration goes behind that below that threshold, then what's going to happen? Well, that's when one reaches profound immunodeficiency. That's the AIDS stage. That's what's going on next. So as there's less and less T helpers, think about it. All right, one is still feeling pretty good as long as they don't get below that threshold. They're feeling okay. During that same period, HIV is slowly but surely creeping up. Slowly but surely. Okay. And now, once you get to about here, now we're going to start to see symptoms and signs when you think of AIDS. So here's phase 3, the profound immunodeficiency or the AIDS stage. This is probably after that chronic latency, which went many, many years. So this might be 5, 10, maybe even longer, 15, 20, depending on the person, what type of medical attention they're getting after the initial infection. The T helper number now goes below that threshold, that asterisk mark. Okay? I think I talk about this in this way. It's like getting closer to the edge of a cliff. As you get closer to the cliff, you're still fine. You're 10 feet away. You're 5 feet away. You're 2 feet. You're still fine. If you turn back, you'd be okay. But once you hit the edge of the cliff, boom, you fall. Same thing happens here. When there's too low of a T helper concentration number in the body, then there's not enough T helpers to do their normal jobs. So you can imagine, normally there are excess T helpers. But when it gets below the T helpers threshold, then there's just not enough. And the jobs aren't being completed. So what's the result? Okay, we know T helpers are the trainers, right? So how would you fill this in? Think about it for a second. You can pause. Okay, I will keep going. Well, this is going to be a T-independent response now, folks. It's T-independent. There will still be plasma Bs being made, but they're fewer and they're weaker because there's not enough T helpers. It's as if there's not enough T or no T helpers. And remember, if there's no T helpers, there can be no cyto-Ts, no new cyto-Ts. So you've got a greatly weakened immunodeficiency. And also think about it, if you don't have T helpers, you can have no memory cells, neither B nor cyto-T. So what's going to happen? Because there is basically a T-independent response now, because there's not enough T helpers to go around, a person will get repeat infections. All right, their body will fight something, but they'll overcome it. But next year they can get the same thing because they didn't get the memory cells. Also because it's a weaker response. There's fewer, weaker plasma Bs. And then no new cyto-Ts. The diseases take a long, long time to recover from. So it takes longer to recover from them because the person doesn't have enough antibodies, B cells, no new cyto-Ts, and they have a tendency to repeat. Once you do get rid of them, they can come back in your body and cause the same problem, same severity, because you don't have the memory cells. So looking at profound immunodeficiency stage, AIDS stage, the number of T helper concentration goes below this threshold. When that happens, look what happens. The HIV number skyrockets. All right? Because there's really pretty much nothing to stop them now. There's not enough cyto-Ts. There's not enough antibodies. And then you're going to get, opportunistic diseases. Think your immune system's greatly weakened now. So even things that weren't really posing a threat to your body, they become problematic. All right? Opportunistic. And so you have a hard time fighting them. And again, you have no memory against them. So you can keep getting them over and over again. What are some examples of diseases that are problematic for a person in the AIDS stage of HIV? Herpes simplex virus. All right? And sarcoma, that's even cancers. So you're like, well, why are these two examples of people that are in the AIDS stage? And this kind of explains why people die of AIDS. The host is unable to fight off these opportunistic infections, number one. All right? Because just a weakened immune system. If you're immunocompromised, latent infections become reactivated. And also, and some examples of things that go latent that once you become immunocompromised can become active. What are some examples? Thinking back. TB, herpes, just some examples. And also, how about cancer? Well, this isn't kind of within the coverage of the class, but to fight cancer, you use your immune system too. So if you have a weakened immune system, your body's more likely to suffer and die from cancer. And like I said, you can get reinfected and diseased by the same agents since there's no memory cells. So you can imagine the person who is the host, all right, the diseased individual is just overwhelmed by all of these things. And finally they die because they're just overwhelmed by them. Well, how about treatment, prevention? Like I said, there is no cure. However, over the last about 30 years, they've come up with a lot of different drugs to either at least slow down the virus. And they're usually given in cocktails, meaning there's several drugs at a time are given to a person. For instance, reverse transcriptase inhibitors, things that just inhibit the reverse transcriptase enzyme, slow it down. Base analogs, those that are targeted mainly for reverse transcriptase. Protease inhibitors. Remember that the HIV protease is very important. It needs to cut up the fusion HIV proteins into individual usable units, like cutting the beer, the cans that are in a six-pack into individual ones so they're useful. And integrase inhibitors. So those are given as cocktails. And for some people, they last for many, many, many years. All right? They say they're diagnosed usually in the chronic latency stage. And that extends their life a long, long, long time before they actually come into the AIDS stage. They can also, if it's diagnosed during the AIDS stage, it can prolong their life too. Now, there's going to be a point where the virus becomes resistant to the drugs. But these have been very successful in extending life. There's no vaccine as of yet because of the high mutation rate, but the search continues to go on, and they're getting closer and closer. In terms of prevention, one that's fairly new since about 2013, I believe, is called the PrEP, or the pre-exposure prophylaxis. And what this is that people that have a very high risk of getting infected by HIV. For instance, sexually active people. Let's say they are prostitutes, sex workers. Let's say someone who has a partner who is HIV positive, and they still want to have sexual relations with that person. It's kind of hard to expect someone to be chaste for 20 years, even if you're devoted to a person. They're given a drug to prevent the chance of being successfully infected by HIV. So here is the person who's not infected, the person who does not want to get infected. They're going to take a drug to prevent getting successfully infected. Another example would be a IV drug abuser. They would also be taking these type of drugs, PrEP drugs. The one that's most famous on the market is called Truvada. All right, here's a little bit of a Truvada ad. All Truvada is, it's just a couple of different base analog medicines, and they target reverse transcriptase. So here's our reverse transcriptase. All right, it shown here in this picture, and usually it takes, again, HIV RNA, and before it can be integrated, it needs to be converted to DNA, right? Converted to DNA. And so here's the human nucleotides. These are in the cell of the human being. Truvada is given. Truvada is here. Is here. All right. It mixes in the reverse transcriptase, can't tell it apart, incorporates Truvada, these base analogs in, and that's a dead end. Okay, it doesn't have a female end, and it stops extension of the new DNA by reverse transcriptase. And that can halt a person from getting infected and diseased. Okay. Just to recap, been very successful. Very, very successful. But the fight goes on against HIV and AIDS. It's been a long battle. Like I said, it started in the 80s. Things have gotten better, but there still isn't a true cure, and there's still no successful vaccine. But I think within the next 10 years there will be. I'm hopeful, at least, of that. Okay, so now let's switch gears to a brand new topic which we'll introduce just briefly today. And so we're going to introduce the topic of fungi. So remember, this is another kingdom. Okay, fungi are eukaryotic, first of all. They're part of the eukaryotic domain, and they're heterotrophs. So heterotrophs means, of course, they're not photosynthetic or chemoautotrophic. They have to steal sugars and things like that. But what else does that just mean? Okay, well, thinking of eukaryotic, that means if it's eukaryote, it's got a nucleus, it's got mitochondria, it's got an ER, Golgi, and so forth. So just remember, eukaryote, it carries a lot with it. And remember, fungi are eukaryotes. Most fungi are saprobes, meaning they are heterotrophic. Again, they're thieves, energy thieves. They feed on dead organic material, like, rotting wood, compost. You'll kind of see them in the forest, all right? Like mushrooms. They're usually breaking down dead things, and that's how they're getting their energetic energy to grow, all right? Now, they're key to recycling. So, without fungi in the world, a lot of things wouldn't break down. Like trees, if fungi went around, they'd be lasting a lot longer in the forest. And even like stuff that, in your garden and things like that, if there wasn't fungi around, things would just kind of accumulate and pile up. And so it's a good thing fungi are around. A little cocktail, even, they have lithium batteries. You think, wow, lithium batteries, like, man, those must last forever. Well, they've actually found some fungi that can actually even break down, very slowly, but will break down lithium batteries. So fungi are really known for the ability to break down and recycle things, and it's really useful and helpful to the earth. So let's talk a little about the good and the bad of fungi. As I've explained before, when we talk about food microbiology, there's many industrial applications of fungi. They're used to make certain kind of foods, like a soy sauce, certain type of cheese, for instance, blue cheese. That blue is from a mold that grows in it. And of course, beer and wine both require yeasts. So there's a lot of good there.
Secondly, which not as known as well, but it's really, really important, is that fungi are like a model experimental system to understand other eukaryotes, human beings. So we say they're a good system for research. So by studying the yeast or a mold, you can learn a lot about what happens in human beings. So we say fundamental biological processes that are studied in simple eukaryotes, like yeasts and molds, can be used to understand humans because there's a lot of similarities between humans and fungi. I'll just give you one real simple example here is you have to see pictures of these children on top here. They have a thing called a Zellweger syndrome, and basically they have problems with their peroxisomes. Peroxisomes are organelles that are found in eukaryotes. You can find them in fungi, and you can find them in human beings. As you can see, these children have afflictions, all right? And sadly enough, they usually die fairly young in life, and they suffer quite a bit. They have Zellweger syndrome, and they realize this, the Zellweger syndrome is due to a defect in peroxisomal function in the human beings. Now, what they found, interestingly, was that the peroxisomes of human beings with Zellweger syndromes, they often can, their defect, they can find the similar defect in yeasts. So they'll say, wow, they can look at a human peroxisomes with a defect in specific PEX genes, peroxisome PEX genes, and they'll say, wow, these yeast cells with these peroxisomal defects look very similar to human cells of people that have Zellweger syndrome. And so what they have found out is interesting. They've learned a lot about peroxisomal genes, those involved in Zellweger syndrome in humans, by studying and understanding how peroxisomes work in yeasts. So you can see it's a very strong system for research and understanding. It's a lot easier to work on yeast cells than it is on human cells. But what you can learn in yeast cells, you can apply to human cells. Also, yeasts don't have cancers, but you can study like growth control genes in yeasts and molds, and again, learn about how they work in human beings. Finally, the bad of fungi. Well, some fungi are parasitic. They cause diseases, and for instance, in plants, animals, and humans, and we'll talk about those in the next lecture.
Well, that's enough lecture for today. We'll talk more about fungi and fungal diseases next lecture. But let's just end it with one kind of dad joke. All right. All right. Well, there it is. Well, take care. Stay safe. See you soon again. Bye bye. And thanks for listening.